The following shows a map of a chromosome in corn. The wild-type alleles are denoted by plus and the mutant alleles by W, S, or E. And we can see that W and S are separated by 8 map units and S and E are separated by 14 map units. And you generate F1 trihybrids by crossing a wild-type, pure breeding wild-type individual to a triple mutant WSC over WSE. And the first question is, if an individual is test crossed, what fraction of the progeny are plus 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 over WSE? The first step is to write down the cross that gives us the trihybrid individual, and therefore we have plus 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 over plus 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 when that's one of the parents and they're being crossed to WSE over WSE, and this will give us a trihybrid individual who is plus 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 over WSE. And we can actually make a map of the chromosomes of this individual. So you will have plus allele, plus allele, plus allele, and the other chromosome will have W allele, S allele, and the E allele. And this trihybrid individual is then test crossed. So we are going to write that test cross, and we have plus 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 over WSE crossed to WSE over WSE. This is our tester, who is homozygous for the recessive allele of all 3 traits. And I brought the map over here. And the question asks, what proportion of the progeny? So the probability of obtaining a plus 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 over WSE individual in the progeny. Now, the WSE chromosome is going to come from the tester, and all their gametes are WSE, and therefore this probability is the same as getting what is the probability of getting a plus 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 chromosome from the trihybrid individual. We note that this plus 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 is a parental genotype, since the trihybrid individual has a plus 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 chromosome. Now, the only way to obtain or preserve the parental genotype is for there to be no crossovers between W and S, and also no crossovers between S and E. Now, what is the probability of a crossover between W and S? W and S are separated by 8 map units, and therefore they produce 8% recombinants, or the probability of crossover is 0.08. What is the probability of crossover between S and E? S and E are separated by 14 map units, which means they produce 14% recombinants. Therefore, the probability of crossover between S and E is 0.08. 0.14. From this, we can compute the probability of no crossover between W and S as 1 minus 0.08. Since you can either have a crossover or you cannot have a crossover, there are only two possibilities, and therefore if 8% of the times you have crossover, then 92% of the times you do not have a crossover. Similarly, the probability of no crossover between S and E is 1 minus 0.14, which is equal to 0.86. Let's make a little bit of space here before we proceed. Okay, and so what we are looking for is there to be no crossover between W and S and no crossover between S and E, and therefore by the product rule, the probability of getting a parental genotype is 0.92 times 0.86, which is equal to 0.7912. Now, there are two parental genotypes, plus, 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 and WSE, and therefore, if you don't have any crossovers and you get parental genotypes, half are going to be plus, 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 and the other half are going to be WSE, and therefore the probability of getting a plus, plus, plus individual is 0.7912 divided by 2, which is 0.3956, or 39.56%. Therefore, the fraction of progeny who are plus, plus, plus over WSE, which is the same as the fraction of progeny resulting from a plus, plus, plus gamete from the trihybrid parent, are 39.56%. In the second part of the problem, we are asked that if the F1 individual is selfed, and so we will write that cross, the F1 individual is plus, plus, plus over WSE, and they're crossed to an individual of the same genotype, then what proportion of the progeny are plus S plus over plus S plus? And in order to get a plus S plus genotype, we must have two crossovers. So there's one crossover here and another crossover in the second location, and this would result in an individual who has a plus S plus genotype. And the probability of a crossover between WNS is 0.08 since WNS are 8 map units apart. Similarly, the probability of a crossover between S and E is 0.14 since S and E are 14 map units apart. Therefore, they will produce 14% recombinance, which translates to a probability of crossover of 0.14. And in order to get a double crossover, and therefore a plus S plus genotype, we must have a crossover between W and S and a crossover between S and E. So we can apply the product rule so that the probability of a double crossover is 0.08 times 0.14, which is 0.0112. Now, every double crossover event leads to two genotypes. There's plus S plus and W plus E, and therefore the probability of getting a plus S plus gamete is half of the probability of a double crossover, which is the same as 0.0056. Now, that's the probability of getting a plus S plus gamete from one of these trihybrid individuals. However, what we have to compute is the probability of getting a progeny who is homozygous for plus S plus, and therefore they must get the plus S plus chromosome from one parent as also the other parent, and therefore by the product rule it would be the product of getting plus S plus squared, and that is 0.0056 times 0.0056, which, if you do the math, is 0.00003136 or 0.003%, and that is very small indeed, which is not surprising since double crossovers are rare events in and of themselves. But here we are asking to have a progeny who has two chromosomes that result from double crossover events, and therefore that's a very, very unlikely event. In the next problem, we are given three traits in mice. There's big W waltzing gait, little w non-waltzing gait, big G gray coat, little g albino, big B bent tail, little b straight tail. A waltzing gray bent tailed mouse is crossed with a non-waltzing albino straight tailed mouse, and the following progeny totals were obtained. We are given these data and we have to determine whether the genes are linked or unlinked, and if genes are linked, we have to determine their map distance. Now, let's consider the different possibilities here. The first possibility is that we are talking about three different unlinked genes. So there are three chromosomes involved, and these are all independent traits assorting independently. If that is the case, then we expect by Mendel's law of independent assortment a 1 is to 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 1, 1, 2, 3, 4, 5, 6, 7, 8. We expect a 1 is to 1 is to 1 or 1/8 frequency for all the combinations of phenotypes. And it's clear that here we are not getting this 1 is to 1 ratio that you would expect by independent assortment, and we can rule out the possibility that all the 3 genes are unlinked and on separate chromosomes. Next, let us consider the possibility that all 3 genes are linked on the same chromosome. So a situation like this. Now we know that in such situations there is a very typical pattern of progeny phenotypes, and that pattern is that the parental genotypes are the most common, followed by single crossover genotypes or phenotypes, and the double crossover genotypes are the least common. And once again, this does not appear to satisfy the pattern of progeny phenotypes, where the parental would be the most common, followed by recombinant genotypes resulting from single crossover events, and finally the smallest number of progeny would be recombinant genotypes resulting from double crossover events. And therefore we can eliminate this possibility as well. And that leaves us with only one other possibility, that 2 of the genes are linked and on one chromosome, and the 3rd gene is unlinked and on a separate chromosome. If that is the situation, then the pattern we expect is that when you're looking at one of the linked genes and the unlinked gene, you expect a 1 is to 1 is to 1 is to 1 ratio, because in fact these 2 genes are unlinked. Also, when we look at the other linked gene along with the unlinked gene, then we will again get a pattern of 1 is to 1 is to 1 is to 1 ratio, because the orange gene is on a separate chromosome and these 2 genes are unlinked as well. And therefore, if 2 genes are linked and on the same chromosome and 1 gene is unlinked, you expect 2 groups of 1 is to 1 is to 1 ratios, and that is indeed what we see here. There is a 1 is to 1 is to 1 is to 1 block here, and there is another 1 is to 1 is to 1 is to 1 block over here, suggesting that 2 of these genes are linked on one chromosome and the 3rd gene is unlinked. Now, the only remaining thing for us to do is to figure out which are the 2 genes that are linked and which gene is unlinked. Now, if we look at this first group of 1 is to 1 is to 1 is to 1 phenotypes, we see that all the possible phenotypic combinations between gray albino G and bent straight B are present in this 1 is to 1 is to 1 ratio, and that suggests to us that these 2 genes are unlinked. So let's say So you could either have G on this chromosome and B on this chromosome, and I will write a different possibility. Let's say B on this chromosome and G on this chromosome. So far, all we know is that they are unlinked, but we don't know which particular chromosome they go to. Now let's look at the other group of 1:1:1:1 ratios. And there we see again that all the possible combinations between waltzing, so W and G, waltzing gray, waltzing albino, non-waltzing gray, non-waltzing albino, are present here in a 1:1:1:1 ratio. And that suggests that W is also unlinked from G. W is unlinked from G, and therefore the only place where I can put W is over here, and that means that this is the only possibility where W and G are unlinked and B and G are also unlinked. In the last step, let us determine the map distance between B and W that are linked. And to do that, we should determine the frequency of the parental genotypes and the frequency of the recombinant genotypes. And so we have the following possibilities. We have waltzing and bent, and then we have non-waltzing and straight, we can have waltzing and straight, and finally we can have non-waltzing and bent. Now we can go and determine the frequencies of these genotypes or these phenotypes. And so let's do waltzing and bent. That's here. And we also have waltzing and bent over here. So we have 39. Then we can look at non-waltzing straight. That's 19 and 22, so that's 41. Then we can look at waltzing straight. That's 9 individuals. And finally, we can look at non-waltzing bent. And that's 11 individuals. Now, we were already told that waltzing and bent were parental genotypes, or the genotypes of one parent, and non-waltzing straight were the genotypes of the other parent. But even if we hadn't been told, just by looking at the frequency, it's clear that waltzing bent and non-waltzing straight are parental genotypes, whereas these guys over here, waltzing straight and non-waltzing bent, are recombinant genotypes. The total number of individuals here is 100, and our percent recombinant is 20 over 100 or 20%. And therefore we can say that B and W are linked on the same chromosome and they are 20 map units apart, whereas G is on a separate chromosome and is unlinked from B and W.